right, thank you, Alon, and thank you, everyone, for being here today. I'm very happy to close out the showcase. So I'm in my 4th year here at Oregon State, and I've spent that time working at the Superfund Research Center. So I'm going to start by telling you about that. It is one of several centers across the country that all study Superfund sites. So what is a Superfund site? Well, it's a heavily contaminated site that is on something called the National Priorities List, which is what it sounds like. It means it's a priority to get them cleaned up. So while these are the centers that study Superfund sites, this is a map of the Superfund sites in the country. There's a lot of them. And they are costly. The average cost of cleanup for just one of these sites is $60 million. So there is a huge cost burden associated with cleaning these sites up, hence the need for the centers to research them. So you'll notice there are several Superfund sites in the state of Oregon, so I wanted to tell you a bit about one that many of you have probably been right around. And that's the Portland Harbor Superfund site. This is a 10-mile stretch of the Willamette River as it leads up to the Columbia, and it is one of the most complex and therefore expensive Superfund sites in the country. Its estimated cost of cleanup is $1 billion, with a B. So how do we as engineers manage a $1 billion problem? Well, we have to break it up into things that are a little bit easier to manage. So what happened at the Portland Harbor is that it was contaminated from decades of heavy industrial use, many of those decades being unregulated. So we ended up with a lot of contaminants in that riverbed. So with preliminary testing, engineers went in and we said, all right, what contaminants are around? And it turns out there's a lot. 64 of them were named contaminants of concern. These are the contaminants that are the most abundant and the most toxic in the site. Therefore, they're our biggest problems. And you'll notice several of them you probably recognize, like heavy metals and pesticides. So 64 is still a very long list, a lot to manage. So that was focused down even more into just 4 contaminants of concern. And the reason why naming these is so important is that now we have concentration goals that become the objective of the remediation project. We need to reach concentration goals that are considered safe for human interaction, and then we can say we're done with the project. So you can kind of think of this like a funnel, where you're taking this very big, complex problem, and you're focusing it down into something that's pretty easy to intellectualize, a concentration goal, reaching that level. So now that we kind of understand that process, I want to show you a problem that arises with that process. And to do that, I'd like to give an example from one of those 4 contaminants of concern at the Superfund site. They also happen to be what I study for my Ph.D. They're polycyclic aromatic hydrocarbons, which is a long phrase, I know, but really all they are compounds with multiple ring structures that have double bonds that are just made up of carbon and hydrogen atoms. That's all it is, and I'll refer to them as PAHs from here on out. So as an engineer, my role is PAH remediation. I try to develop technologies to clean up the PAHs from the environment. And specifically, I work in bioremediation. So I use bacteria to do the work for me. As Jesse told you earlier, bacteria are really great chemists. They're also really great cleaners. So that's what I use them for. So if you think of bacteria as like little Pac-Men, and then the PAHs as those pellets that the Pac-Men eat, the bacteria really do eat the PAHs, and then they produce a waste product afterwards. So I'm interested in 3 different aspects of this process. One, are the PAHs being removed? That's a very important question, typically the only question that we're asking in remediation studies. But I'm interested in a few other things. What are these bacteria doing during this process? How are they eating the PAHs? And also, what's that waste that's being produced? Is that something we should be worried about? So to give you an example of why those latter 2 questions are so important, I want to share a story from my research. So this is a plate of a pure culture that I work with in lab. It's a nice bright orange color, so go bees. And these are some chemical structures of a few of the PAHs that I study. So in my experiments, I just mix these 2 together in little batch bottles. I take samples over time, and I take some measurements to try to figure out what's going on with the PAHs and with the bacteria. So going back to those 3 questions that are important for my research, from one of our more recent studies that we did with that pure culture, I asked, are PAHs being removed? And we found out, well, yes. We actually got 75% removal of the PAHs in just 5 days, which is a very short treatment time. So we were really pumped about that. And then what these bacteria are doing, well, they're partially eating the PAHs, and they're staying alive in those 5 days, which is great, because ideally I'd like them to work longer than just 5 days. But that waste that's being produced, well, turns out it's twice as toxic as the PAHs we started out with, which is a big problem, right? I don't want to make things more toxic by cleaning them up. And as a reminder, typically that first question is the only one we're asking in remediation studies. So from that aspect, it looks like it's a great remediation strategy. So that brings me to this final question in my presentation title of, are we helping or hurting? I showed you that we have a remediation technology that I've been working on that is great at removing PAHs. So if you're looking at things from that approach of funneling down your problem into just those contaminants of concern concentrations, we're doing a great job. But because we kind of broadened our problem in our line of questioning back out, and we said, well, we're starting with a toxic site, wouldn't it be great if it was less toxic afterwards? We kind of find out that this remediation strategy probably shouldn't be used in the field, and it won't, don't worry, we're not putting this in the Portland Harbor. So this is a really relevant finding, not just for engineering practice, but also engineering research. A recent review that came out from our center looked at almost 300 PAH remediation studies, and we found that, well, 100% of them are looking at those PAH concentrations and trying to reduce them, as they should, but less than 2% were looking at post-remediation toxicity. So we're all operating under this assumption that if we're reducing those contaminant of concern concentrations, that then we're reducing the toxicity, which is often, but not always true. So in my work, as I'm developing remediation strategies, I'm trying to look at the whole problem, not just that simplified one, so that I can really be helping the problem, not hurting. Thank you.